In the last video, we talked about motivation. And now we're going to talk about a particular motivation, one of the sort of basic physiological motives, and that is hunger. The goal of hunger is actually eating behavior. That's the goal of what we need to do if we're hungry. And it's influenced by several different things. It's influenced by the biological process. It's influenced by psychological processes. And it's also influenced by social, cultural influences. So we can't understand hunger if we just look at biology. We need to understand the context in which hunger occurs, the psychological influences, and the social, cultural influences. Hunger is a great example of homeostasis, the fact that we need to have mechanisms that actually encourage us to start eating and processes that encourage us to stop eating. Remember Dieter's fly that we talked about in week 1? It had a mechanism, hunger, when it tastes with its feet to start eating, and it had this nerve that went from the esophagus of the fly to its brain that said to stop eating when food was present in that area of the body. It's a combination of starting and stopping. Homeostasis, keeping things at the steady state. Hunger, and so what we need to understand with these basic physiological motivations, really all motivation, is that we need to have something that starts us to do something, but we also need to have something to actually stop us from doing something, in this case, eating. The homeostatic mechanisms for hunger are located in the brain, the stomach, and the liver, and we're going to talk about all three of those. The feedback, when do we eat and when do we stop eating? Those are the two processes that we need to understand. Now first, biological influences is the stomach. If we had food in the stomach, then we are not hungry, and if we don't have food in the stomach, then we have hunger. They're called hunger pangs, and you've all experienced that. It's interesting to note that it's not volume of stuff, it's the calories that are generated by the food. For example, if we eat a big giant bowl of celery, then big giant bowl of celery or a small piece of chocolate cake, the small piece of chocolate cake will take longer to leave the stomach than will the big bowl of celery because it doesn't have many calories. So going through the stomach depends upon the calories that it generates. We have hunger pangs, this feeling of hunger, and it's correlated with stomach contractions. A classic study done by Cannon and Washburn in 1912, a century ago, was to actually measure the contractions of the stomach. And what they did was they put a balloon down into the stomach, and so that balloon, and blew it up, so this balloon can measure the contractions of the stomach. And it had the subject, the person with the balloon in their stomach, to simply say, press this button whenever you have a hunger pang. And what they found was that the feelings of hunger were correlated with stomach contractions. So stomach contractions tell us that we're hungry and want us to start eating. Hopefully not a balloon, but food. But the classic study is showing these correlations. And there also, the brain is important in producing hunger. And it really occurs in the hypothalamus. It's part of the brain and the limbic system that we talked about when we talked about the brain. The hypothalamus has many different parts, and I've sort of demonstrated them here. There's the posterior area of the hypothalamus, the ventromedial area, the anterior area, the medial preoptic area, and the lateral area. And the areas of the hypothalamus that are important for hunger are the ventromedial area and the lateral area. The interesting thing is that the lateral area is the part of the brain that tells us to start eating, and the ventromedial area tells us to stop eating. So research that's been done with animals, for example, if they lesion the lateral area, they won't eat. There's a famous film that shows a cat, little small kitten with no lateral hypothalamus, and they put a mouse right in front of it. It's very hungry, and it won't eat. They have to be force-fed. And the ventromedial area is for stopping eating. And if you lesion the ventromedial area, the animal will eat and will not stop eating because it doesn't have the ventromedial hypothalamus, and it will get very, very big. Like Dieter's fly, that just simply got very, very big because it did not have the inhibition that was necessary to stop eating. So the lateral area of the hypothalamus is to start, and the ventromedial area of the hypothalamus is to stop. Again, that positive feedback, negative feedback, that's a part of homeostasis. Now, this is called the dual center theory, and it was very popular in the 60s and 70s, but it's really has some problems with it. First, we know that it's much too simple. We've already shown you that the stomach itself is important in producing hunger and satiation, and we're going to talk in a minute about the liver, the third part. So it's just too simple. It only deals with one aspect of what produces hunger. Lesions to both areas also cut, if you lesion the ventromedial hypothalamus or the lateral hypothalamus, it really also cuts the tracts going through the hypothalamus. So it's doing more than just in that particular area of the brain. And both seem to affect metabolism. So even if the ventromedial animals receive a normal amount of food, they still gain weight. So it seems to affect metabolism as well as just the initiation and cessation of feeding behavior. And finally, lateral hypothalamus lesioned animals show reduced drive generally and other motives. So it's important. Obviously, the hypothalamus is critical in understanding hunger, but just using that as the start and stop mechanism is actually much too simple. Now, there's also some debate about what the hypothalamus actually does, what it's measuring, what is the sensor mechanism that's producing the behavior. Some believe that hypothalamus monitors glucose, and others believe that the hypothalamus monitors lipids or fats. Both are important in determining what hunger is, but which one, we don't really know. We talked about the stomach, we talked about the brain. Now let's talk about the liver, the third biological process that is important to understand what hunger is all about. The liver monitors the balance of glucose, which is usable blood sugar, the blood sugar that's in our blood and ready to produce energy, and glycogen, which is stored blood sugar, stored in our fat. And it monitors that, and when glucose rises, the pancreas, it tells the pancreas gland to release insulin, which causes the cells to store the excess glucose, suppressing hunger and storing it in fat. And when glucose lowers, the pancreas releases another glucagon, which causes cells to convert the stored glucogen into glucose that we can use. So it's taking the glucose and storing it if we don't need it, and if we do need it, turning it into glucose from our fat that we can use. And that's what the liver does. It's this monitoring mechanism that tells the pancreas gland what to do. So hunger is produced by a high ratio of glucagon to insulin. So the liver is another monitor, like the hypothalamus, like the stomach, that causes us to understand what hunger is all about physiologically. There are also psychological factors. The memory of our last meal. We tend to avoid unfamiliar foods. If it's something we don't know what it really is like, we tend not to want to eat it. I can remember as a child growing up in the South that we would have brains and eggs for breakfast. Now, only in certain areas did you do that. The brains came out of a can. They were all chopped up so you couldn't tell what they were, and I happened to like those. And then I went to New Zealand for a conference one time, and I went to a restaurant, and they had lamb's brains on the menu. And I said, God, I just love that. I used to eat it for breakfast with eggs when I was a child. And so I ordered them. And what came out on a plate were two intact, perfectly shaped brains. It looked like the brain that I showed you when we talked about the brain and behavior. I had a hard time eating that. It just looked too much like a brain. I liked it when it was all chopped up and mixed with eggs, but I just had a hard time eating it when it really was a brain. I think you would too, most of you. So the sight of food is important, and the smell of food is important. When I was a child, I probably would not have eaten Camembert cheese because it smells so awful, but now I know it's really delicious. Stress, if we're stressful and we have certain kinds of moods, that affects our hunger. Also, whether we're healthy and feel good affects whether we want to eat. And then we've already talked about, when we talked about learning, conditioned aversions. Where, for example, I won't eat raisins. I picked them out of foods because I got sick while eating raisins when I was a child. So there are many psychological factors that affect our hunger also, and our feeding behavior. And there are also cultural factors. For example, taste preferences exist across culture. In certain cultures, the eye of a camel is a really preferred food. I don't think I want to eat the eye of a camel if I knew what it was. And it's also interesting that if you live in a culture that has high temperature, you tend to like spicy foods. Here's a graph that shows that, the mean annual temperature of a particular culture and the number of spices that you would typically find in a menu. For example, this would be Finland, which the average number of spices in food is 2. And this would be India, a much hotter climate, and the average number of spices in their food is 10. So cultural factors, psychological factors, and certainly biological factors are important in understanding hunger. Thank you.